Okay, so I'm going to go ahead and get started. Hi, everyone. Welcome to the third webinar in the Community Science LI webinar series. We are so excited to have you all joining us today. My name is Ariel Santos, and I will be moderating today's webinar. For those of you who are new to Community Science LI, the goal of this series is to highlight various community science opportunities around Long Island and how the data accumulated by the public influences local environmental management efforts. This project was created in partnership between CTUC Environmental Association and Long Island Sound Study. CTUC is a nonprofit 501c3 organization dedicated to conserving Long Island wildlife and the environment. Long Island Sound Study is a cooperative effort involving researchers, regulators, user groups, and other concerned organizations and individuals to protect and improve the health of Long Island Sound. Together with support from the Peconic Estuary Partnership and the South Shore Estuary Reserve, we bring you Community Science LI. So just some housekeeping notes, though I'm sure you are all more than familiar with how this works at this point. This webinar and all future webinars are being recorded. All of the recordings will be available on CTUC's website shortly after the webinar concludes. Your video and microphones are turned off, but you are free to ask questions in the Q&A section at the bottom of your screen. You are more than welcome to utilize the chat box as well to add any comments or say hello, of course, but we will be monitoring that Q&A closely to answer any questions during the presentations and after during the dedicated Q&A period. With that, I would like to introduce the panelists for today's webinar. We have Jimena Perez Vizcasillas, Long Island Sound Study Outreach Coordinator and co-host of Community Science LI. She will be paying close attention to the Q&A and helping me moderate today. We have Casey Pendergast and Samantha Hoff, wildlife technicians from the New York State Department of Environmental Conservation, who will cover bat ecology and natural history of bats on Long Island. Emily Hall, conservation policy advocate from CTUC, who will go over BatMap LI survey and how data accumulated from the survey is used. And finally, myself, Ariel Santos, policy program coordinator, also of CTUC. And I am the other co-host of Community Science LI alongside Jimena. And as I mentioned earlier, I will be moderating today's webinar. So without further ado, I will go ahead and let Casey and Sam take it away.
All right, thank you. Okay, can you see my slides?
Looks good, Sam.
Good, all right, thank you. Welcome everybody. Glad to hear that there are so many people interested in hearing a bit about bats. So I just want to give a brief overview first of the bat species we have present in New York, and then specifically some more details about the ones we can find on Long Island. A little bit of background about white nose syndrome and how that influenced the creation of our research project on Long Island, and then dive into what our research entails and a little bit about what we hope to do with all of this information we've gathered up to this point. So there are 9 species of bats found in New York, all insectivorous, so they all eat insects. And we have 3 tree bat, or sometimes called migratory species, the hoary bat, red bat, silver-haired bat. And then we have 6 cave bat species, or our hibernators. So they hibernate locally in the state in either caves or mines, or some other structures, as we might be finding on Long Island. So we have little brown bats, northern long-eared bats, Indiana bat, eastern small-footed bat, tricolored bats, and big brown bats. So the 2 that are in black there, Indiana bat and eastern small-footed bat, have never been present on Long Island, so I'm not going to go into any more detail about those 2, but just wanted to mention they are found in the rest of the state. So these 3 are our tree bats, or migratory bats. So they do seasonally migrate. Typically they arrive in the spring and leave in late August, or later in the fall, sometimes as well. So hoary bat, red bat, and silver-haired bat, fairly common in the summer. We do detect them pretty frequently with our acoustic surveys, and we've also captured some during our netting. There have been some interesting accounts of silver-haired bats popping up earlier than we would expect, even throughout the winter. So it's possible that some hang around, but the majority of them do seem to migrate further south for the winter. And there's not a ton known about migration of tree bats. It's kind of an ongoing research effort, and it doesn't seem to be as straightforward as bird migration. So they don't just, head straight to where they're going to go spend the winter. Sometimes they kind of circle around and zigzag back and forth and then end up, wherever they end up for the winter. So. Okay. Yeah, so for our hibernating species, they hibernate essentially because of the changing seasons and changing environment. So it's related to their physiology. The majority will spend the summer roosting in trees, using these structures in the spring and the fall as well. But during the winter, they need a temperature stable environment, so either a cave or a mine. And this will stay near freezing temperatures, but above freezing. So they enter what's called torpor, where they lower their metabolic rate, essentially just, shutting down their immune system and metabolic rate at sort of a basal level. And occasionally they do arouse from torpor, so they're not just, hibernating or sleeping the entire time. They do have these periodic arousals, but for the most part, sort of in a period of rest. And again, this is largely because there's no food available on the landscape during the winter for our insectivorous bats. The energy source for hibernation is fat. So typically in the fall, they do what's called swarming behavior. So they're flying around, feeding as much as possible to gain that body fat that's going to help them sustain energy throughout the winter, essentially to account for those periodic arousals of torpor. So our first hibernator, the little brown bat, historically was found on Long Island, and they were formerly the most common species throughout New York in pre-white nose syndrome years. After white nose, declines of about 83% throughout the state. They do like to hibernate in clusters or occasionally as single individuals. And then in the summer, they do form large maternity colonies when they're raising their pups or their offspring. There is some recent evidence of population stabilization within New York, and this comes from a few different research studies that have been conducted for this species, partially from our hibernation counts that we do every winter, and also some genetic studies as well. So maybe some good news for the little browns. Unfortunately, there's been no captures of little brown bats on Long Island in post-white nose years. And in all the acoustic studies that we do, we rarely ever detect them across Suffolk County. So unfortunately, the population hasn't rebounded down there yet. Oh. And in terms of identifying them, they are a little brown bat, of course, but they do have slightly different, coloration between their belly, which is a little whiter, and their back, which is brown. Next, we have northern long-eared bats. Pretty wide distribution. They were, fairly common, but never in huge numbers before white nose. But throughout most of the state, declined 99% since white nose arrived. They are federally listed as threatened, and this happened in April 2015. And there is a petition to have them uplisted, essentially to endangered. So that review is ongoing. They can be a little difficult to monitor because they do like to hibernate as solo individuals, and they often like to crawl into little cracks and crevices, so they're really hard to count with our traditional, monitoring efforts. But we do still find them in large numbers in across Long Island, but particularly Suffolk County. So I will talk much more about that later. Identifying factor here, they look very similar to little brown bats, and that can be difficult if, unless you have them in hand. The one main feature is the little tragus in their ear is much longer than it is for little brown bats. Tricolored bats were never, very common in New York State. It is kind of the northern portion of their range. Unfortunately, declines up to 97% in New York State. And they are also currently being reviewed for listed under the Endangered Species Act as well. Do typically roost or hibernate in caves and mines in warmer areas, and they do like to roost alone as well. They have been detected across Long Island, of course in low numbers as well, but we do know of one maternity colony and one hibernation site that were discovered in recent years, so that is a good sign. In terms of how to identify them, they because they're called tricolored bats, they have these 3 different colorations. They're kind of blonde fur with a red forearm and a black wing. Yeah. Okay. Big brown bats, currently our most common species in New York. They can be a little difficult to monitor with our traditional methods as well, because a large portion of the population does spend the winter in human structures, mostly things like attics or barns or garages, depending on the temperature. So if you do find a bat in your house, often it is a big brown bat, but Long Island, that might not be the case. They do like to hibernate alone, rarely in clusters, and they're our largest cave bat, so they are bigger than the other species I just discussed. And again, year-round found on Long Island. And we also tend to get some information from submissions to rabies labs. So if somebody does find one in their living space and it gets submitted, that also gives us a good source of information of distribution. And characteristic feature, they have this kind of longer, glossier fur and like a bulldog-looking nose compared to our other smaller cave bat species. So white nose syndrome, what is it? This is a very bat-specific disease caused by an invasive fungal pathogen, Pseudogymnoascus destructions, or PD for short, because that's a mouthful. And it's very bat-specific because it grows at essentially the ideal temperatures that bats like to hibernate at. So low temperatures, high humidities, essentially creates the perfect storm for this fungus to infect bats when they're hibernating. It currently only goes through a fungal asexual reproductive cycle, so there's not any diversity in the strains that have been found in the United States yet. Unfortunately, it does remain in the environment in hibernacula, essentially indefinitely as far as we know. It can hang out as a spore and just wait for a bat to come back in the fall and reinfect them. And it was first discovered in New York, Albany and Schoharie counties, in a few of the caves that we have around here in the winter of 2006, 2007. And it's mostly spread from bat to bat once they become infected, particularly for our species that like to cluster in the winter. They're going to share those fungal spores and infect each other. And in the initial years, it caused, a large amount of mortality. Some other symptoms we observe are wing damage, because the fungus does actually grow into the wing membranes and cause, erosion and holes and things like that. Unfortunately, its major mechanism to cause mortality is that it affects those natural torpor cycles throughout the winter. So the bats would naturally wake up every few weeks, but because the fungus is kind of an irritant and it also can cause dehydration, it causes them to wake up more frequently than they normally would. If they do go out on the landscape in the winter, there's nothing for them to eat and they can starve. And this is just an image, also what my background is, but looking at a bat wing that's infected under UV light, you can actually see fluorescence of the areas of wing damage. So it's an important tool for detecting, an individual that's been infected, but also for quantifying that infection as well. So unfortunately, this has had, negative implications for a lot of bat species nationwide. It now affects, I think, up to 13 species it's been detected on, essentially. Numbers, killed over 6 million bats at this point. That's kind of an old estimate, so it's probably, millions more at this point. But species are affected differently. So some are not experiencing as severe infections as others. Some seem completely unaffected. So there is some differences between species. And unfortunately, it just keeps continuing to be spread further and further and is now almost across the entire US. I'll show you a map in a little bit. It did come from Europe or Asia. It's been hard to detect the actual source, but closest identification of a strain was likely in Western Europe. The bats there do show visible infection with fungus, but they don't have the associated mortality we've seen here. So it kind of implies that it's been around for a while there and they've either adapted or evolved to tolerate an infection. So good sign for the European bats, but not for ours, unless a similar thing can happen here. Two of the species that seem to be less affected are two that we have in New York, so small-footed bats and big brown bats. And of course, bats play an important role ecologically. So having this massive mortality from this invasive disease is going to affect all these important services that we get from bats. They play a really important role in pest control, so eating insects that are pests of forests or our agricultural crops. There's been a few different estimates of how much money that actually saves by bats enacting this service, and it's about 23 billion estimated savings in the US alone just from these services. There are bats that pollinate as well, not the ones we have in New York, but further into the Southwest, so that's another role that they play. And unfortunately, bats are long-lived, so having this effect on the population can have really long-lasting effects. They only have 1 to 2 offspring or pups per year, so it's a really slow recovery time if they are infected. So there's a lot of research going into how white nose might affect reproduction and survival in the future. So just looking at spread of white nose, this is, it's going very quickly. The first year, 2007, kind of localized. Next year, spread quite extensively until we're where we are essentially today, where it's been found all the way out in on the West Coast in Washington and down into Texas as well. And you can see this kind of follows the karst layers or available caves based on topography throughout the U.S. It was thought that maybe the Rockies would be a barrier, but that doesn't seem to be the case either. And it does just continue to spread. And we update, these maps with more and more counties every year. So moving to focus on northern long-eared bats, which is the study of our subject. This map is just showing you their total distribution. So they are found out into the Midwest and throughout the Southeast a little bit and into Canada. But unfortunately, white nose has now, covered their entire range. So there's no area where we find northerns that is free from white nose at this point. And there's been numerous studies that have predicted this could lead to extinction for northern long-eared bats. So just looking at certain sites, depending on pre-white nose syndrome colony size, large amount have been predicted to go extinct. And kind of into the future, it remains likely probability of local extinction. So local extinction being, sites like caves and mines. But overall, it's not a good outlook for this species in the future. So I'll talk about, some of the evidence we first found, but I just wanted to highlight some of the goals of our research on Long Island. We've been interested in determining where these bats are found, what their activity patterns look like year round, but then especially in the fall and the winter where they need to be building up that fat to survive hibernation, and where they might be spending the winter locally because there are no caves and mines found on Long Island, and being able to characterize those sites. How prevalent is the fungus that causes white nose on the island? Is there anything available for them to eat? We know the climate is different on Long Island than it is to upstate. You have this more mild coastal climate. So would that actually facilitate their ability to eat in the winter? And then looking at genetic structure as well. But I'm just going to focus on the two that are highlighted here. So these two objectives we use acoustic surveys for quite extensively. So we just wanted to kind of dive into those more deeply. And unfortunately, the history of northern long-eared bats on Long Island is pretty minimal. So the first record was documented in 1933. It was actually a bat in a house. And then this study of mammals of Long Island done by the State Museum was really the only study we have in pre-white nose years. They found a bunch of individuals that they caught misnetting out on the South Fork, basically in Montauk. We do have some information from the rabies lab. So again, we get submissions when bats are found in houses if they might have had contact with a human. And those were always steady in pre-white nose years. And then our first case of white nose was in Brookhaven. Was a northern long-eared bat that was found on the winter landscape that eventually died. And that was in 2011. But in pre-white nose years, we've had quite a bit more evidence that our population is persisting, where it's declined drastically upstate. So we really need to understand why and what's influencing that. And the first method we wanted to use to do this was just understanding, what's the distribution like on Long Island, and are there any associations with certain habitats that might be influencing this distribution? So to do this, we set up a large-scale acoustic study across Suffolk County, and we picked out 50 grids. They're all the little colored boxes here. These are all 1 kilometer by 1 kilometer squares that we identified. And we split these up into different canopy cover levels. So that's what the different colors represent. Northern long-eared bats are typically called a forest interior species. So they're often found in more dense forests. That's where they like to forage and roost. But we were curious if we would find them across other levels of canopy cover as well, and just see, overall what's affecting the distribution here. So within each grid cell, we put 3 acoustic detectors that we would survey anywhere from 3 nights to 8 nights throughout the summer time period. And we did this in 2017, 2018, and 2020. So we wanted to go over a bit of the details of how you actually perform acoustic surveys and what it's like, designing them and looking at files. So Casey's going to talk a little bit about acoustics specifically.
Yeah, so I came into the DEC working with bats, actually working on the stratified grid square design that Sam set up for Long Island. So that was my first experience. And I kind of fell into all this and had to learn on the go. And the basic building blocks of learning how to work with bat acoustics is understanding how echolocation works, how bat calls work. So the bats send out their biosonar, looking for an object. When it hits the object, it bounces back, and that pulse back is what they're hearing to identify the distance between. So when we're looking to record bat acoustics, we're looking at a way to pick up the longest piece of this, pulses that they're sending out in order to identify the species. You can skip to the next slide, Sam. And so when we're doing that, there's a lot of things we have to consider as a researcher. You're looking at different bat species flying all throughout the air. You're looking at the bats' variation from night to night, depending on the temperatures and the environment. And you're getting differences between different bat detectors recording different calls. So you really have to determine which species you're looking for when you're setting up your study. And then even within the site, you have different bats occupying different habitats. So even tricolored bats would be very up high. We have our northern long-eared bats that do prefer to be a little bit lower within a forest interior. And where you place your detector is also determined by trying to have the optimum quality calls. So when you have a detector very close to the ground, you're going to get some echoes from the ground, which would make it harder to identify pulses. Near vegetation is going to create some disturbance as well, some echoes that's going to throw off your pulse. And then it's kind of this, you have to find a sweet spot between a high activity area, like over a water, an open stream would be great. You're getting a lot of bats feeding, but then you're getting a lot of different bat species. And that can lead to mixed calls, making it harder to identify any individual species in and of itself. Next slide, Sam. So when we're looking at bat pulses, we're looking at the whole sequence altogether. We can break it up into 3 different parts. So the search phase is bats spend their time primarily in this phase when they're flying around searching for prey items. These are the even space pulses, about 100 milliseconds apart. And then the approach phase is what we see when a bat detects a prey item or even just something it's curious about. So if you're placing your microphone in the middle of an open area, you'll actually get a lot of approach phases because the bats are curious about this new object that kind of appeared in the sky. And that's where they emit pulses much closer together, almost like the Doppler effect, trying to gauge how close they are to it, to hone in on it. And then the feeding buzz is going to be a very rapid and steep pulses that essentially drop off once they feed on the prey, and then it'll resume back to a search phase. So because bats have very similar calls altogether, all the different species, we are only looking at search phase pulses to identify any individual species. So as easy as acoustics may seem, it actually gets somewhat complicated when you break it down like this. So if we're looking just to kind of go over this in a visual sense, the search phase here is the first third of this call. You can see it's well-spaced pulses. But if you look through the whole call as we go, you can see how much it changes throughout. So in the middle that's our approach. The bat has identified an item and it's honing in on it. And then it gets very, very narrow there. That's the sudden dip down in frequency. And that's the feeding buzz. So you can see this, if you're only getting a portion of this call, maybe you only get the approach or the feeding buzz, you might be confused about what species you're looking at. So we get tons and tons, gigabytes of files that Sam and I have to go through of acoustic files. And you're always searching for the search phase to use as an identifiable piece of the call. Just a quick little visual view of the different bat pulses that are associated with each species we discussed. So they do range in frequency. That's the left-hand axis there on the x-axis, or the y-axis is going to be your frequency. And you can see we have like the hoary, the big brown bat. Those are much lower in frequency, and that's coming back to where they like to forage and how slowly they fly. That's how quickly their pulses are coming out and back. Whereas the northern, you see that towards the middle, it's a very, very steep, narrow pulse. And that's spreading over a large range of frequencies. And that's because northern long-eared bats, they are the forest interior species that Sam mentioned. So they're sending out quick pulses constantly because they're flying through cluttered environments, trying to identify prey in this very narrow area. And then this all comes down to the echolocation signature. So as I was saying, it's the bat's shape of their wings and the weight of the bat is what determines, their ability to fly in a certain habitat. So for the northern long-eared bats, they do have the long wings that allow them to fly very quickly in this cluttered environment, and that relates to their echolocation signature that we just looked at, or the pulses that we looked at. All right, Sam.
Okay. So what did we find in our 3 years of surveying all across Suffolk County? The little orange squares are where we had presence, and the black is where we had no presence of northern long-eared bats for each year. We did have reduced survey effort in 2020 just due to staffing and budget issues, but we were still able to cover almost half of our grid cells here. So the numbers on the right, this is, how many actual detector sites we had presence at compared to the total, and how many cells we surveyed that year. And these other numbers here are the results of some of the modeling I do. It's called occupancy modeling. You don't have to pay attention to too much to what these symbols mean here. I'll explain a little bit, but this means occupancy and this means detection rate. So the trends we've seen over the years are actually our occupancy, the top number has been increasing. So we're seeing presence at more and more sites, but our detection rate is going down. So it means they're getting harder and harder to detect. And there could be certain variables at play that we can then look at through modeling later, like differences in the weather for that night or the time that we surveyed them throughout the summer. But it could also mean that there is a reduction in density. So, we're not getting population numbers from acoustics, but it still can tell us population trends, or we can infer that from the results we're getting. So the other thing I look at is, what environmental associations might be influencing their distribution. And really the big one that we find, kind of no surprise, on Long Island is development. But we do see a difference between high and low levels of development. So high is classified as anything 50% and over, that impervious surfaces essentially, or low development would be 50% and under. And you can see a really steep drop off as soon as, the area we survey is surrounded by a lot of high intensity development, even if it's, 40% or more of the surrounding area, we're much less likely to detect them. Whereas they don't seem to be affected as much with the lower intensity development. And this could be because, you have a lot of fragmented habitat, forested patches across Long Island, but as you move further east, those patches are a little bit closer together, and they might not be as affected by this kind of lower level of fragmentation. You also have larger lot sizes, lots that do have, some forested habitat, and we do find them in really small patches, like even as low as an acre, particularly if we move closer to the east. So overall, the further east we go, especially the forks, the more activity and more presence sites we have for northern long-eared bats.
So that brings it into the research that I was doing. Just a quick background on the northern long-eared bat's diet. It's kind of looked at as a moth strategist as well as an opportunistic forager. So their diet's mainly dominated by beetles and moths, but they have been documented feeding on a wide range of insects. For that reason, if we're looking to study the bat diet, we have to try and appeal to a wide range of insects that we can try and capture with our surveys. So I chose to set up survey sugar bait traps and UV light traps, which could attract, flying insects as well as crawling if they can crawl from the branches. But what we're doing is setting up 2 surveys per month, and that's due to the high seasonality of insects. So they're kind of follow these seasonal activity patterns. So you'll get insects that are active for a very short period in the winter that don't come out for the, until the summer again, or insects that appear in the spring, depending on whether the weather warms. So you're trying to catch these insects in these small windows when they may be active. The historical literature stated that insects were active starting at about 5 degrees Celsius would be the lowest temperature you'd be able to see them. So we set that as our survey requirements, to have at least 5 degrees Celsius 2 hours after sunset when insect activity would be greatest. And in our first year, we did actually find that even below 5 degrees Celsius, because we did have some volunteers that were willing to survey below our minimum standards, they were still getting capturing insects well below 5 degrees Celsius. So there were insects out in the landscape across a whole range of temperatures, but we did stick to the 5 degrees Celsius going forward for at least an 80% likelihood of capturing our insects. And then just here's a quick comparison of our insect activity versus the northern long-eared bat activity for the 2018-2019. So you're seeing highly similar seasonal activity patterns, both in the fall and the winter. In the winter and the spring. You do see on the northern long-eared bat, the mysi activity graph, that they're actually active a bit earlier in March at a higher rate than you would for the insects. But it is important to note that the insect surveys were only occurring, up to twice as infrequently as twice a month, whereas the bat detectors are operating constantly. So it's not the most accurate comparison across the board. We are seeing very similar activity patterns. So the bats and the insects do seem to be out on the landscape at approximately the same time. And then just looking at the activity of the specific insects we've captured. So we're getting fall moths on the landscape well into March and even April, which is something we hadn't expected to see, but in the coastal climate of Long Island, where it is a little bit more temperate, you're more likely to see these insects on the landscape year round versus dropping off in January, February, which was what we would expect to see upstate. And then the Hypena scabra, this is actually an interesting one because it'll come back later when we're talking about guano, but this species was one of our most common species, along with the moth on the last page, the spring cankerworm, collecting on nearly every site. Oops, sorry, Sam.
Oh, did you want me to?
No, you're fine.
Okay.
So the Hypena scabra we're collecting on nearly every site across all of our island regions. We did capture this Hypena scabra in guano in the fall, but it is a spring moth, so we're seeing it active essentially from fall all the way through spring, so year round. Some more spring moths that are active for nearly the entire season. And then these guys are especially curious. These are migrants, so they were coming up from the south, and they're following the frost line. So usually you wouldn't expect to see them until maybe May or June, but we're actually seeing them much earlier in the year than we have before. And it's kind of might be going into the global warming aspect of things, but it's just more proof that Long Island has a climate that's allowing for higher insect activity than you would see elsewhere throughout the northern long bat range. And then this is looking at our temperature variation and insect activity. So as I mentioned, there's a mark there on the graph, the 5 degrees Celsius, which was the historical threshold set out in the literature. And you'll see all the little black dots across there are insect activity that we recorded in our surveys. So that's well below the 5 degrees Celsius line. And then the stars up there are going to be the feeding buzz events. So if you remember when I was talking about acoustics, we can identify when a bat is feeding. So they go through the search, approach, and then the actual feeding buzz phase. So when we see feeding buzz, we're actually finding acoustic files like that throughout the wintertime. So we can prove that bats are feeding on the landscape. And the majority of these feeding buzz events did occur just outside our known hibernacula on Long Island. And then this is just looking at that hibernacula again, just to kind of double down on this the insect activity related to the bats. So we have the bottom graph is going to be inside the hibernacula, and the upper graph is outside. So the scale on the mysi axis is much different. You're at 40 files versus the lower axis is only 6. So we're having significantly more files outside the hibernacula than inside, but all the stars are denoting occurrences where the bat was recorded both inside and outside the hibernacula. indicating that they are emerging at that point. And for 63% of our emergent events on this graph, we are actually detecting insects at the same time outside. So the majority of the time the bats are emerging due to their normal arousals, we are detecting insects on the landscape as well. And then that brings us to our next aspect of it. We've confirmed that the bats are outside, the insects are active during similar periods. They're both emerging below 5 degrees Celsius. The last question is whether the bats are feeding, and is it enough to sustain them throughout their white nose syndrome arousals. So we're looking at guano collection. And to do this, the easiest way would be on the left-hand side, we got a little bat hanging there in the mist net. So when we do capture the bats in our mist netting, we'll store them in a brown paper bag until they defecate. We can use that sample. Normally, in a traditional guano study, you could collect guano from the underneath roosts or underneath hibernating bats throughout the winter. In our study, if you can see the middle there, we have a tiny little bat sitting on a floor joist in a crawl space. This is from our Long Island hibernacula. And that was seen once in one of our many visits to the hibernation site. As many times as we went in there, we had never seen him other than that was the only time we saw him out. So it's a little bit more difficult to collect guano when it's just a single bat living in a large crawl space. And he's only entering and exiting through a small little door there. So it wasn't really proper locations to collect winter guano. So if you flip to the next slide, Sam, you'll see our sampling distribution. We got a lot more guano samples collected in the fall and the spring than we did in the winter, which was our target period. But what was able to help us establish a dietary study for the bat in general, so we can see what they're eating year round. And if you're looking at the full guano analysis here, the fall was actually our most diverse period. So we have 8 of 11 orders were detected in the fall, including spiders, which had been found quite a bit in northern long-eared bat studies elsewhere. And we did find them in their fall, as well as wasps and bees. Throughout the entire study, the Lepidoptera, moths, and diptera flies were the most common, which is, we had heard that they were a moth strategist and an opportunistic forager. So this kind of falls in with what we do know about northern long-eared bats, and leads us on to our management implications, Sam.
Yeah, I know we're over our time. This is my last slide, so don't worry. But what are we going to do with all this information is really kind of the next step in our research. There are regulations on things like a tree that a bat has been roosting in, so you can protect the habitat around there. So it's always important to try to find those type of sites, but we do now have bats, hibernating in human structures on the island. So our next step is really what we can do about that. We're looking into the possibility of creating our own hibernation sites that we could protect, that would be on protected property. The other thing that, because they are federally listed, there are some regulations in terms of things like tree cutting, if you are near an occupied site. So there are some federal, regulations in place, but all of our work is really supporting the time period that those actions can happen, because we do know that we have bats active in the winter and much later in the fall and earlier in the spring than you would expect in other regions throughout the range. So it's really been an accumulation of kind of putting all of our information together and seeing what we can do about it. So that is it. And I will stop sharing and can answer any questions later.
All right. Can everyone hear me okay? Okay, awesome. Thanks, Sam and Casey, so much for that great presentation. I am going to touch a little bit today, kind of just following up on what Casey and Sam talked about on the community science aspect of things. So CTUC, in coordination with Maria Brown and some other local partners, have developed our community science program, Bat Map Long Island. And it's basically an additional survey, kind of as we've been presenting throughout this community science series, where interested bat enthusiasts, volunteers, anyone really can go out just in their local backyard and tell us where they've been observing bats. So I'll go over a little bit more about Bat Map Long Island, how to access it, and then how to input the data. Okay, so as I mentioned, BatMap Long Island was created by Maria Brown, a CTech friend, bat researcher, and teacher. She's a lecturer at Stony Brook University and a science research teacher at Sayville High School. The goal of BatMap is to better understand kind of where bats are on Long Island. You know, as Casey and Sam were mentioning, it's really good to have a better understanding of their distribution, what species are where, how they're using the habitat. So are there foraging areas? Are there roost sites? And then all this data and information helps us to better make conservation management decisions. So like we're saying, you know, is there a certain area that needs to be protected, or do we need to, you know, make some other landscape arrangements to better help the species? So that's kind of the long-term goals of BatMap. All right, and then how do we actually survey? So as you were hearing, bats kind of may slowly start emerging now or kind of migrating now in March as the weather starts to warm, but they'll really begin moving and feeding in spring, so April and May. They will then continue to be active in the summer, and then they'll go back into hibernation around the fall, so October, November. The best times to go out are around dusk or dawn, an hour after or before sunset, so kind of in that twilight phase. All you need to do for this survey is kind of just look around in your backyard, in your neighborhood, or even maybe a local park, if they allow you to go kind of around dusk or dawn. And then as you were hearing from Sam and Casey too, the bats may be emerging from a building, they may be emerging from a woodsy area, they may be in that woodsy area, or they may be moving to more open space or near a water body, such as a pond, even a backyard pool, to hunt for insects. So these are all kind of the tips on first kind of how to survey. All right, I'm going to stop sharing those two, and then I'm going to transfer over into actually accessing the survey. Okay. All right, so there's multiple ways to reach the survey. The first thing that you're going to want to do is go to ctux.org on our website. You can navigate to the menu, go to Get Involved. Under that will be Community Science and then BatMap Long Island. Okay. We have a little background information as well as our contact information. If you have any additional questions, you're welcome to get in touch with us about the survey or about anything related to BatMap. All right. The first location we have the survey is actually right embedded into our website. So you're welcome to even just go to this web page and enter all of your information and data right there. Or you can do it a couple different ways. One way is you can download the Survey 123 mobile fields app. So as you can see, I did that here already. It's that green little box with the check mark, Survey 123. Next, you can either scan the QR code with your camera and that'll bring up a link, or you can just press this link right here, the ArcGIS link. That'll give you an additional two options. One of them is to open in browser. And again, you have access to the survey and it's right within your browser. If you prefer this option, maybe it's just a little bit too squished on our webpage or something, you're welcome to use it this way. Or you can say open in the Survey 123 field app, but you can only do this after you download the Survey 123 app. So that's the first step. And then you go to this link and that's the second step. You can click on this open in Survey 123. It asks you if you want to open this page, you say open, and then it'll bring you to the app. The most important part of this is that you do not need an ArcGIS or Survey 123 account or login to get into the survey. All you need to do is say continue without signing in. Okay, and a few of the things we ask you. We ask you the observation date. We ask you the time that bats were first observed at your location. So this might not be the time that you're kind of entering in your information, but it might be You kind of just have to think to yourself or mark down the time when you see your first bat. The observation location, and this should automatically generate if you have your location services on. If not, or if it looks like you're in the wrong area, all you have to do is you can just zoom in into this platform, find out where you are, and then log it there. You can also just click the plus mark on the side and the minus mark to zoom in and out. And then the mark with the little crosshairs at the bottom will take you back to your actual location. Then all you do is click the check mark at the bottom and your location is saved. The other questions we ask you are the county of observation. We ask you the street location, the city, the zip code, and then we ask you some bat-related questions. Choose the best description for the location where you observe bats. So were they flying overhead? Were they emerging from trees, emerging from a structure, flying over water, as I mentioned earlier? Did you observe the bats at dusk? Yes or no? Bats were observed flying above or at tree line. So they were high flying or they were low flying, or maybe you couldn't tell. As Casey and Sam were mentioning, there's different species that like to fly at kind of different points in the tree line. The estimated number of individuals observed, 0 to 5, more than 5, more than 15. Have you observed bats at this location previously? Is there a bathhouse at this location? And then you can upload some images. Some of the last questions are also, would you be willing to provide access to the observed location for bioacoustical monitoring? So as Casey and Sam were mentioning, bioacoustical monitoring can help kind of get an additional assessment of the bat species that may be present there, their level of activity, different things like that. So we'll actually come to your property possibly and do some more acoustical monitoring so we can get a little bit more idea of what's going on with the bats there. You can say yes or no, or if it's not your property, you can just say other. And then if you are willing to be contacted about your observation or about bioacoustically monitoring the property, you can just put your email down there. All right, you can just click the check mark when you're done. Obviously, I haven't entered any information, but that'll submit your survey. Another couple things you could do is you can click the top X mark there. And if you want to save your survey for later, you can do that. So let's say you're out somewhere and the service isn't great, you can save it in drafts and finish it later. You can go ahead and continue the survey, or you can just close it and lose your changes. And I wanted to bring you back to this main My Survey 123 page because it has all the different surveys on it. So you have to upload each individually, but it's a great way to go in and see all of the different community science surveys that are right there at your fingertips, and you don't have to kind of switch between pages or platforms. All right, and then I just wanted to show you again how to get back into Survey 123. You just go to the app again, you click without signing in, and the bat map in the whole page is right there. Okay. All right, I'm gonna stop sharing now. Okay, a couple other things I just wanted to mention quick before we got to questions is that for all the attendees who registered today, I'll add everyone onto our BatMap Long Island Outreach listserv so that we just kind of periodically update you on any other volunteer opportunities, just kind of anything happening with the monitoring season, anything interesting that we're seeing. We'll just kind of keep you up to date on some of that stuff. If you don't want to be on that listserv, that's totally fine. You can just email me at ehall@ctalk.org, or you can just unsubscribe from the email when you get it. And then additionally, we have some other cool bat-related events that you may or may not be familiar with. So that includes Bats and Brews. It's our annual fundraiser focused around bats and local craft beer. But we often have a really awesome bat speaker come and just talk about, a different research topic involving bats. So you're kind of welcome to look into that as well. That information's on our website. And we often share some cool bat facts as well on our Bats and Brews Instagram and Facebook. So you can check those out too. All right, so Ariel, I'll then pass it back to you for any questions.
Awesome. Thank you so much, Casey, Sam, Emily. That was great. It looks like we've pretty much answered all the questions in the Q&A and in the chat. I do have two here. that we can get to. From Karen, she has a question. I have a bat in my yard regularly. Should I report them every time I see them? Emily, I don't know if you want to take that one.
Sure. I would say, yeah, you can report it as often as you'd like on BatMap. I mean, maybe you see it at different times of the day. Maybe you can notice different aspects of its behavior. Definitely, I would at least get it into the BatMap survey, and then we can start kind of communicating with you on what you're seeing. So yeah, definitely more observations the better, I would say, in terms of BatMap.
Awesome, thank you. Then I have a question from William. Any hints on identifying bats from small birds at dusk? I don't know if Sam or Casey, you want to tackle that one?
Yeah. Hey, Bill. That's a tough one. I struggle with that as well. I feel like typically birds are like on a mission at that point. It's dusk. They're trying to get back to where they're going to spend the night. I think bats more just from their flight pattern kind of flutter around a bit more and are kind of checking things out and looking for insects. So sometimes that helps, but if you don't get a good view, if it's too quick, I mean, it's tough.
The mechanics of bat flight versus bird flight are so different that it's the jerky motions of the bat flying that make it a little bit obvious that it's a bat versus a bird. Not that I am a professional in identifying the two, but yeah, it's a very different mechanical aspect behind it. So it kind of, if you think about it that way, which one looks smoother, and if you do a little background on birds versus bats and how they can fly, it'll actually help you out a lot there.
Awesome, thank you. And I think you touched on this in the chat box, but Patricia has asked, how does one ID insect orders in guano samples?
Yeah, I see.
So go ahead.
I saw Sam responded to that pretty clearly, but it is a very simple DNA analysis procedure. Not that it's the most exact by any means. A number of the samples are, only come back to order, and quite a few actually we were able to get down to species. So the chart I showed only went up to order, but that was, I did have to throw out, but remove from that graph a number of species or samples. They only got down to they could just tell that it was an insect and that's it. So not the most exact, but a lot easier, a lot more simple than the old-fashioned morphological analysis where you're just under a microscope breaking apart guano pellets and trying to identify parts of moths and beetles and stuff from there.
Also, not that we did the DNA extraction or analysis either. We sent that out to be done at another university because neither one of us are genetics experts, but yeah.
Thank you. I have another question from Dave. Can you speak more about moth species in the woods locally in early spring? Also, I've noted a good number of slant-lined moth. Forgot the science name, he said. Are these moth fodder as well?
Sorry, I didn't quite hear the start of that one. You could repeat it, sorry.
Yeah, no problem. Dave just asked, can you speak more about moth species in the woods locally in early spring? And he noted a good number of slant-lined moths. Are these moth fodder as well?
Yeah, so there's a large number of species that are actually active in the spring, more so than the fall. The fall is kind of a very limited breed, the fall moths and the winter moths, whereas the spring, that's when, especially Long Island, that is an agricultural hotspot out there on the East End. You get a huge number of species that are attracted to the sap that you can get from the fruit trees. So you'll get this huge influx of moths that are going to be coming in the springtime as soon as the buds start to come out and the frost line recedes. And even in my study, I wish I'd put it up there, but I have a species list, and I think it's over like 40 or 50 species long just from our insect study of different moth species. So in this, this is in the times of the insect Armageddon, right? Where we're reading all the time about insect die-offs that they're just finally able to track. So there are quite a few species that I don't know, and I can't talk at length about a slant-lined moth. But there are a large number of species that you'll see in the spring, and a good number of them are kind of pest species that are feeding on the sap trees, the sugar of sap trees.
Awesome, thank you. And he also asked, he's seen red bats near him pretty relatively often. Are they common in Western Suffolk?
Yes, definitely. We don't do too much mist netting out that way because we're usually out on the east end where we've captured them there. But based on our acoustics, they're all over Suffolk and very common. And I always like to say the red bats are taking over because it's like the majority of the files that we look at are all red bats. But yeah, very common.
And just like Long Island serves as this migratory pathway for birds heading, migrating down the East Coast, the same is true for bats. So you get a huge number of red bats that are migrating from further north down, the same with the hories and the silver-haired. And you'll see them, you'll see big peaks of activity during the migratory times. But we, as Sam's commenting, we do see them year-round throughout the region, so.
Great. And then I have a question from Lisa. Are you planning on doing any future mist netting, and are volunteers able to attend?
Yeah, our netting period is probably over. um, yeah, we're both almost finished with our graduate degree, which is kind of concluding this project. At least the netting and tracking portion that we do. We'd like to keep doing acoustic surveys because we think it's an important management tool. So that's certainly something, especially with CTUC, getting involved in that as well, that volunteers could help with. Yeah, I think.
Awesome. And yeah, so after this concludes, everyone who has attended today, you'll receive an email that has everyone's, email information. So if you have some specific questions about how you can get involved, you'll be able to contact someone. I do see Mike Wittini. If you want to ask a question, I believe I can unmute you. Give me 1 second. Okay. Mike, you should be allowed to talk.
Yeah, it looks like. Thanks, Casey and Sam. It was great. And yeah, I've always been curious. Well, I have like a million questions, but I'll just give you this one that has always been curious to me. Yeah, I'm familiar with Connor's work, and I noticed that he did a lot of his mist netting over small ponds. And I don't know if it's the nature of the mist nets in terms of bats versus birds, but I was always wondering why he wouldn't go out and you guys as well. How come nobody looks at the salt marsh where there's all these insects out at night? And is it a matter of it's just too open to catch the bats, or? I
Yeah.
would think, I would think there would be a bat specialist that just does the salt marsh.
Yeah, we've used acoustic detectors like kind of on the edge and gotten a lot of activity. But it can be difficult without to net in those environments. Typically we're trying to put a net on a narrow trail where they're kind of funneled into our net. So something that has like a low enough canopy and is kind of dense on the sides. We haven't ever had good luck netting over ponds, which we have tried, and we've tried to replicate at the ponds he netted over on Hither Hills, and we never seem to have luck. And it could be that, the density is lower anyway, because he just reported, droves of bats, flying around and just flying into his nets. And we've never had that experience. We've caught some birds that way, of course, but that was unintended. So yeah, we're typically trying to find spots we can net where the bats are going to be kind of funneled into the net, and then they do like water sources, and we've caught a bunch over even like puddles and ruts in trails. So that's been useful. But yeah, as Mike just commented as well, Mike Fishman, also the wind can be tough on the marshes.
Okay.
Yeah, so they're pretty good at actually seeing the net if it's moving, or even if it's like foggy and there's a little condensation on the net.
Right.
They can pick that up with their echolocation.
Yeah, we've seen bats. Even we're standing off the trail and we've seen a bat flying full speed right at our net at the last second, swerves and avoids it. So they're very capable of avoiding these nets when they're in the trail fully kind of funneled into it. So when they're out in the open like that, it's a lot more difficult. And part of it is it's the species that we're looking for. So we're looking for northern long-eared bats that would be a forest interior species, and they wouldn't necessarily be found foraging in an open area like a salt marsh.
Gotcha. Now, the last question is, I'm monitoring an eagle nest for the DEC, and see, 3 nights ago, right at dusk, I had a bat fly up and down the road, right over my car. I was sitting in the car, and I actually managed to get a look at it with my binoculars. Is it possible to ID a red bat by the reddish tinge without actually, having it in hand? Is there anything that has a bit of a reddish color to it?
Yeah, it most likely is a red bat. From all the accounts that people ever tell us or submit to us, if they actually get like a photo of one or they see one flying, like even during the day, it always ends up being a red bat. The tricoloreds are more of a blondish color, so there's really nothing else you could confuse with a red bat if you are seeing a good like red fur color.
Okay.
Yeah, it's probably most likely one.
All right. Thank you.
Yeah, thanks, Mike.
Thanks, Mike.
Thank you,
I'm
Mike.
gonna mute you again.
Okay, let's see. I think that is all of the questions we had, right? If you have any further questions, like I said, we will be sending out an email with all of our contact information. So it is 4:04. To respect everyone's time, we'll go ahead and wrap up the webinar for today. I just want to take a moment to thank our panelists, Casey, Sam, and Emily. Thank you so much for sharing your time and your knowledge with us today. I know I learned a lot, and I'm sure all of our participants did as well. Thank you, Jimena, my co-host and co-creator of Community Science LI, for helping me moderate today and keeping track of all those great questions we received. And finally, thank you all for joining us to learn more about bats on Long Island, how we can monitor them, and how that information we gather as volunteer community scientists help protect their populations. If you have any questions that weren't answered today, like I said, please feel free to reach out to us. With that follow-up information, our contact information will be on there. All the links to the resources we shared will be on there, in addition to the link to view the recorded webinar. And that'll be up shortly after we conclude today. So again, thank you all, and we hope you can join us for the 4th installment of Community Science LI. That will be on April 13th, which will cover horseshoe crab monitorings. So I hope you guys enjoy the rest of your day. Bye, everyone.
Thank you.